Hello, everyone. Thank you for joining. We will start in about 1 minute. I am very much looking forward to today's presentation by Steve Mistead, presenting on Equashield's role in hazardous drug containment. It is going to be a special presentation that Steve has been working on for a bit of time, pulling together, is it decades, Steve, of experience about?
Unfortunately, yeah.
No.
Showing my age. Showing my age.
No, it's fortunate for all the attendees today for sure. So we will get started in just about 1 minute. And again, for those of you who've arrived early, thank you very much. We really do appreciate it, and we will start on time and end on time today. And I will just explain some of the mechanics of Zoom for those of you who may not have used it, familiar. In the window that you're watching, you can expand it to make sure it's full view on your screen so that you can see everything. And then secondarily, if you move your mouse cursor down to near the bottom, you will see a chat and a Q&A. They're both enabled, but Q&A is really where you want to put your questions for Steve to answer. The chat scroll is just so hard to navigate, go back through to figure out who asked what, that we're ask you to put your Q&A into the Q&A box, and then those questions will be fielded by Steve, time permitting, post. So that just one quick thing before we turn it over to you, Steve, if I could ask all of you who have signed in already to, in the chat box, type in where you're dialing in from today. I have the fortune of being right there back at home in Green Bay, Wisconsin. I see Fred's in Nebraska, Michigan, Seattle. Oh, awesome. All over the country, Steve. This is looking good. Fantastic. Chastity from Mobile, John in St. Paul, Ashley here in New Jersey. Fantastic. So Steve, with this, Ricky Thackeray, fantastic, with Equashield is on the call today as well. Wonderful. So Steve, I'm going to turn it over to you, and let's get your presentation underway.
Okay. Well, first I want to thank Equashield for allowing me to present on this important topic, and also Pharmacy Stars for providing us the platform. This is a really important topic, I think, and we're going to discuss today hazardous drug containment, but primarily we're going to be looking at the closed system transfer device, or the CSTD, and how that helps us keep contamination under control. So first, what is a hazardous drug? Well, NIOSH, the National Institute of Occupational Safety and Health, describes a hazardous drug as one to possess one or more of the following. They look to see if it's carcinogenic, teratogenic, if it has a reproductive toxicity, if it's genotoxic, or if it has toxicity, organ toxicity at low doses, or if the molecular structure or toxicity profile is similar to a known hazardous drug. We've known for decades that employees are, at risk for exposure to hazardous drugs. This can come in a mass exposure, such as a drug spill, but also there are many different areas in the hospital or a facility that handles hazardous drugs that have contamination throughout the facility. And even these minute contaminations build up into the system and can address and come about to the healthcare worker in acute and chronic conditions, rashes, reproductive problems, infertility, malformation of fetus, spontaneous abortion, cancers, and even death. There are three major routes of exposure for the healthcare worker. They're dermal, inhalation, and ingestion. The dermal route is, of course, from direct exposure or direct contact of that drug. But also, we have to, again, think about these places of contamination in our facility. They're going to be places that are high-touched areas, like biological safety cabinets, countertops, floors, equipment, tables and chairs, and really any place or most places in an area where hazardous drugs are handled or administered. And I think there's a study out there that even shows that with the proper engineering controls and proper PPE and actually the deactivation and decontamination processes in place, we still have this contamination through our facility. So the key here is to ensure that we decrease our contamination in our environment. The route of inhalation can be, of course, from suspended particles in the air from traditional compounding or reconstitution of vials and that sort of thing. But also we have to be really aware of these certain drugs that are highly volatile at room temperature, such as cisplatin, ifosfamide, cyclophosphamide, etoposide, 5-FU, just to name a couple. And it's important to remember this because when people are manipulating these drugs, especially in the administration side, they're exposed to potential vapors that could be harmful to their health. Then ingestion, same thing. We have the particles in the air that can settle down on food and beverages, but also as healthcare professionals, we often become too relaxed around hazardous drugs or anything when we get familiar with it, and we tend to skip our processes and procedures. And many people might forget to wash their hands after handling a hazardous drug, and you get that direct contact from hand to mouth causing ingestion. So where does a closed system transfer device come into play here? NIOSH defines a closed system transfer device as a drug transfer device that mechanically prohibits the transfer of environmental contaminants into the system and the escape of hazardous drug or vapor concentrations outside the system. It's a known fact that CSTDs have shown to decrease hazardous drug contamination in our environment and exposure to our employees. For compounding, although a CSTD is not required, because we have the biological safety cabinet, our engineering controls keeping us safe if they're used effectively and appropriately, which again, fatigue can play a role in that. But I'd like to move us beyond that, saying that instead of just saying it's strongly recommended, actually considering it a requirement. Because one, when we are manipulating something in the biological safety cabinet, we're causing contamination. Everything coming out of that cabinet is going to be contaminated and start that snowball effect of contaminating all the areas in our hospital or our facility, starting from compounding to the checking process to administration and even waste. So decreasing, again, our potential for contamination is going to decrease our exposure to our employees. Then we need to think about administration. We want to give the end user who is administering this drug the full closed system. If we're not using a CSTD, essentially we deliver our final preparation to the person administering, and they have to go through manipulations to secure a CSTD, which is required for anti-neoplastics, essentially exposing themselves. So we want to give them everything they need. They have nothing in between them and the product besides PPE. So they really need that extra line of defense of a CSTD. Then when we talk about disposal, we don't really think about that a lot because once it's out of sight, out of mind, we throw it away, it's gone. But in fact, if it's sitting there until it's picked up, you could have potential vapors escaping from that waste bin, and you get exposure to nurses, to patients on different therapies, even visitors visiting the patients, and EVS workers who are cleaning around that area. Next, we want to look at a traditional syringe and look at the exposure areas from that. The traditional syringe, often CSTDs attached to this traditional syringe, with the exception of Equashield. So let's look at this a little bit. So the barrel, inside that barrel, once that drug is pulled back, you have contamination inside of the interior of the barrel. And in addition to that, you have the back of the syringe that's fully open, so you can get escape of chemical residues as well as vapors exiting that end of the syringe because it's fully open, nothing's stopping it. Then you have the plunger. The plunger is actually using the side walls of that syringe to support itself. So it's in contact with it. It's becoming contaminated as well with hazardous drug residues. So if pulled back, whether it's delivered to a floor for a push or something you're sitting down in your biological safety cabinet, those areas become contaminated. As well, if you accidentally touch it with your hand, then your gloved hand becomes contaminated with hazardous drugs as well. And then because of that closed back, or I'm sorry, that open back of this traditional syringe, the plunger can easily dislodge from the back, causing a drug spill. Then if you move to the tip of the syringe where the needle attaches, this area, if you're not using a CSTD, you get potential contact of inhalation through vapors and contact through exposure through contact of the actual product or liquid itself. There's a peer-reviewed study out there that's fairly recent about showing the inner walls and their contamination. This study uses an open back syringe like we just saw with a CSTD attached. It uses 3 drugs, 5-FU, cyclophosphamide, and I can't say this, ifosfamide. And they do 15 transfers, simple transfers, simply of the drug to the syringe, then the syringe to the bag. It's a very uncomplicated transfer. And they do this 15 times with different syringes for each drug. They use Chemoglow HD clean wipes, where they swab the interior of the barrels after the fact to see what kind of contamination was collected. The results were that there was significant hazardous drug contamination found on the inner walls of the barrel of the syringe. And also they found that the contamination between the 3 drugs were not statistically significant, showing that potentially any drug could act the same and cause contamination. Here are the results from that study showing each of the 3 drugs and the syringes used and those manipulations and associated contamination with each one. You'll note that the cyclophosphamide looks a little bit less contaminated, but the cyclophosphamide was a 20 milligram per mil versus the other 2 were 50 mg per mil. And that could be meaning that there's a concentration potential making something more or less contaminated, but also we have to think about the quality or the drug dynamics and how they interact with the syringe walls, or maybe a different affinity for different drugs. So other drugs may even cause more contamination than we see here. So the authors concluded that the levels of contamination found inside these syringes were of concern and could be potential exposure to the environment and as well as healthcare workers. Also, they said that we need to really identify ways to limit contamination and exposure in order to keep our people safe. So in STEP's Equashield, we want to look at Equashield because they've employed some special technologies that really answered some of these questions that the definition that NIOSH has out there that really brings into place what NIOSH is intending for a closed system transfer device. These technologies are a closed barrel, a centered plunger, a welded connector, enclosed needles, dual membrane to membrane connection, and this pressure equalization that's pretty spectacular in my opinion. The closed barrel is actually what it says. It's that black disc in the back is closing that barrel so nothing can get out and nothing can get in. It's essentially containing any chemicals or hazardous drug residues as well as vapors inside that syringe. Additionally, it provides a safety net because that plunger cannot be dislodged from the back of the barrel, causing a drug spill. Then you have the centered plunger, which you'll note in the picture that it doesn't come in contact with the sidewalls. Therefore, it doesn't become contaminated with hazardous drug residues. So if set down on a surface or accidentally touched by your gloved hand, you're not getting contamination transfer. And then in addition to that, if you have microbial growth on your hand and you accidentally touch that plunger, when you do your manipulations, it decreases the potential of microbial contamination getting into the syringe and into your final preparation. And then because of this closed system and this disc and the plunger not being able to be dislodged, the FDA actually clears this to be used for every single increment on the syringe. You can pull back and use this full volume of the syringe instead of this 2/3 rule that we always hear. And this allows for better measurement because we know using a smaller syringe is always better for better accuracy, and we are dealing with chemo. And depending on your manipulations, you actually could get fewer manipulations out of your compounding session. And then there's also economical and environmental help with this because, as we know, larger syringes can cost more than smaller syringes. And also when we dispose of those syringes, we fill up our waste bin quicker and then we have more disposal, which costs a lot of money. So we can save some money there as well as some environmental issues. There's a vapor containment study that's out there that took Equashield and compared it to some competitors. And the study was conducted by Nebraska Methodist Medical Center. They used the NIOSH test, proposed NIOSH test for IPA. They wanted to look at the worst case scenario since it's highly volatile to see if these CSTDs could actually contain vapor. The failure rate was indicated by the detection of greater than 1 part per million detected in the air. It was chosen as 1 part per million because that was the amount that could be detected accurately. That was a lower limit, what was accurate. They knew they could measure at least 1 part per million. Only 2 CSTDs passed, one being Equashield. Then another study was also done by UNC that actually showed these same results. Let's talk about the welded connector a little bit. It's pretty awesome advancement, I think, because it's one connection. It doesn't come off, so you prevent accidental disconnections during compounding or administration. And also, it prevents alteration after dispensing. The last thing we want to do is for a nurse or a person who's administering this drug to get a hold of it and administer it and have to remove it because they don't know how to use it. We need our employees trained so they can be safe, and this would prevent any kind of alteration of the syringe. Additionally, it's welded. There's no glues or adhesives. So there's one polypropylene pathway, so we know our compatibility. And also because there's less pieces, we save time and it's more efficient for us to use. We also have a dual membrane to membrane system. This membrane to membrane system, once they come together and connect, the needles will pierce those membranes so you can do your alterations and your manipulations. But once you remove that connection, the needles retract into the membrane and are wiped clean, keeping a dry surface. So it has no leakage or wetness for where contamination could occur. It's a total dry disconnect. And also because those needles are inside the membranes, that you can't get any potential needle sticks. There's a study out there by UNC that shows the dry disconnect. It's by UNC. They use litmus paper and 5FU to detect whether or not there's a wet disconnect. And they showed that only the membrane to membrane systems, one being Equashield, had a dry disconnect. All other non-membrane to membrane CSTDs in the study showed wetness or leakage on litmus paper. Nebraska Methodist Medical Center did the same study and confirmed UNC findings, but actually found that Equashield was the only one that had a true dry disconnect. The competitor membrane to membrane system in the other study did very well, but it did show a slight wet disconnection. Then the pressure equalization. This is something I find pretty amazing as well, and it all is involved because that's closed system. There's 2 needles in this system. One is for air, one is for liquid, and it's very simple. You connect the 2 pieces, you start your manipulation, and air goes back and forth in liquid. There's no thought processes. It's just really easy and simple to and intuitive to use. In addition to that, because there's sterile air at the back of the syringe, it's closed, nothing's getting in, nothing's getting out. Once those connections are made, you have a true closed connection or closed system. So we know that reducing hazardous drug contamination and occupational exposure is critical to decreasing adverse health effects to our healthcare workers and others in contact with these areas. Equashield has 100% reduction of surface contamination, showed by peer-reviewed studies, as well as a 0% contamination of the plunger. They show vapor containment through a proposed NIOSH protocol using IPA. They have 100% dry disconnect, and also they're shown to prevent microbial ingress into the system. The FDA actually clears this system up to 10 entries, so you can attach a vial connector or a vial adapter to a vial and actually go into it 10 times per the FDA. So we're going to watch a couple videos. They're very short. I think they're less than a minute each. This first one is on preparing a simple syringe, whether it's for dispensing or for further compounding.
Align red to red and engage syringe unit to vial assembly. Invert vial and withdraw required amount of drug. Inject air bubbles into vial and continue to withdraw until the required amount is obtained. To reduce volume, turn vial in its upright position and inject the drug into vial. Disengage syringe unit from vial assembly. Syringe unit is now ready for administration or downstream preparation. Dispose of or store components per facility protocol.
All right, the next video we want to look at is actually preparing a bag using the same syringe we just prepared in the previous video. I just want to note that the bag adapter used here, you'll see that there's not a secondary line. I'm thinking that Equashield did this because there are different policies, protocols out there depending on where you work that says who should be priming lines and that kind of thing. My opinion, it would be best practice to always prime a line prior to doing any kind of manipulation to the final preparation and before sending it out. So again, they have everything they need and it's all ready to go. But just keep in mind, you can take a secondary or primary line and attach it to this adapter, or Equashield even has another solution where they provide their own secondary set, so you can choose either or.
Using a spike adapter, spike an IV bag up to the ridge on the spike. Engage syringe unit to spike adapter and inject drug. Flush spike adapter by aspirating at least 0.5 milliliters and re-inject. Disengage syringe unit from spike adapter. IV bag is now ready for administration or downstream preparation. Dispose of or store components per facility protocol.
Okay, so those videos were, I think, very beneficial just to get a quick introduction of how Equashield works. But again, let's revisit this definition by NIOSH about a closed system transfer device. Again, it's a transfer device that mechanically prohibits the transfer of environmental contamination into the system and the escape of hazardous drug or vapors outside the system. And I don't know about you, but I often forget about this ingress of contamination, trying to prevent that. When I think of CSTD, I'm always thinking about, let's make sure it's contained and nothing gets out. But it's important to remember that NIOSH definition actually says both. We're trying to prevent the ingress into the syringe and also the egress out of the syringe. And you can see that Equashield really provides us a lot of benefit than any other CSTD out there, with a closed back that's totally closed, a suspended plunger or rod, a closed or a welded connector that can't be removed, and it makes it very easy to use with the pressure equalization. So the question comes up, why are there so many CSTDs on the market? Well, the FDA has this thing called the predicate device approval system, and they don't really approve CSTDs, but they clear them based on the fact whether or not there is a CSTD out there that is similar to the one you're trying to introduce. So as we know, though, not all CSTDs are made the same. The membrane-to-membrane systems seem to work better, Equashield being one of them, but Equashield is the only one with a closed back and that centered plunger. Also, we have those lure-based systems that they just leave wet disconnects, and the filter-based systems, they can't contain vapor at all. So Equashield really comes into alignment with this definition of NIOSH and the CSTD. Again, that's closed back allows for prevention of ingress or egress of any contamination, and also prevents the accidental dislodgement of the barrel, and it allows the FDA to say, go ahead and use this for its full volume. Also, there's that centered plunger that again prevents the ingress or egress of any contamination, because that plunger does not touch the walls of the inner barrel of the syringe. And again, no other CSTD offers this advantage. Then you have the membrane to membrane connection that offers a dry disconnect. preventing the egress of any kind of contamination, and also prevents needle sticks. You have the syringe unit itself that's welded together as one connection that prevents the egress of hazardous drugs and also prevents accidental disconnection and alteration after it's dispensed, and also makes you more efficient because it minimizes pieces in time. Then we have that equalization, pressure equalization. Again, with two needles, easily being able to manipulate with the fluid and the liquid and the air going back and forth being sterile makes it very intuitive and easy to use, which I include that as a safety feature because it's just the thought process is so easy. You don't have to think at all. You just start and you manipulate. Again, no other CSTD really offers these advantages of the closed back and the centered plunger. We know the lure-based systems have a wet disconnect. The filter-based systems can't filter out vapors. And all these other CSTDs out there actually use that open back syringe with the plunger that can be contaminated to connect. Only Equashield has the solution, I think, for closing that system so you are protected 100% as an employee. So how do you choose the right solution for you and your facility? Well, you need to carefully evaluate the claims of any CSTD manufacturer. You want to look at the claims and make sure they're backed by peer-reviewed and independent studies, and ensure the methods used in those studies are actually quality. They show efficacy. You want to choose a CSTD that aligns with the NIOSH definition. You want to make sure vapors cannot escape. You want to have that dry disconnect. And you want to make sure your hands and surfaces aren't becoming contaminated, as well as preventing that ingress of microbial contamination. So I think we can see how Equashield really aligns and actually exceeds this NIOSH definition, and something that probably should be considered in most facilities for the safety of your environment and your employees.